Hi everybody, and welcome to our module on folate and vitamin B12. In this module we're going to discuss folate, which is also sometimes called vitamin B9, and vitamin B12 together. So why are we talking about these two vitamins together? Well, that's because they're both used for the synthesis of the nucleotide thymidine, which is used to synthesize DNA, and they are also both used in the metabolism of homocysteine. This means that if you're deficient in either vitamin, you can get some similar symptoms. If you're deficient in either vitamin, you will have less DNA synthesis because you cannot form thymidine. This leads to a megaloblastic anemia, which we'll talk about in this module. And in addition, if you're deficient in either of these vitamins, you will have an increased level of homocysteine in the plasma, and that's because you cannot metabolize the amino acid homocysteine. And I've shown you on the screen here the structures of thymidine and homocysteine to remind you what they look like. Synthesis of the nucleic acid thymidine is complex and multi-layered, and it involves both vitamin B12 and folate. So we're going to walk through it slowly on this slide so that you can understand the role that the two vitamins play. So the first thing you should know is that the nucleotide thymidine monophosphate is synthesized from the nucleotide uridine monophosphate via the enzyme thymidylate synthase. And uridine monophosphate and thymidine monophosphate have almost the same structure. The only difference is that there is one extra carbon added to uridine to create a molecule of thymidine. This means that the synthesis pathway is going to involve transferring this one carbon to uridine so that you can synthesize thymidine. That one carbon comes from a molecule of N5-N10 tetrahydrofolate. This is a molecule that is derived from the vitamin folate. In donating that one carbon to uridine, N5-N10 tetrahydrofolate becomes dihydrofolate. And so you need to keep regenerating N5-N10 tetrahydrofolate from dihydrofolate if you want to continue to synthesize thymidine inside of cells. Dihydrofolate can be converted back into N5-N10 tetrahydrofolate via the enzyme dihydrofolate reductase. It converts dihydrofolate into tetrahydrofolate, and then tetrahydrofolate is converted back into N5-N10 tetrahydrofolate. So this cycle keeps regenerating a constant pool of N5-N10 tetrahydrofolate that is available to synthesize more molecules of thymidine, and you need the vitamin folate in your cells in order to make this whole pathway occur. The way that vitamin B12 fits into this pathway is that N5-N10 tetrahydrofolate can be converted into N5-methyl tetrahydrofolate, and then the methyl tetrahydrofolate is regenerated back into tetrahydrofolate by vitamin B12. So if you lack vitamin B12, all your folate will get tied up or trapped as N5-methyl tetrahydrofolate, and you won't have enough of this structure around in order to synthesize thymidine. The last piece of this complex pathway involves the amino acid homocysteine. Homocysteine is converted into methionine in the process of regenerating tetrahydrofolate by vitamin B12, and we'll talk more about how that occurs in a minute. Let me clear away all my ink here and show you the big picture. If you lack folate in cells in your body, you will not be able to convert uridine into thymidine because this process of reactions will shut down. Additionally, if you lack vitamin B12 in your cells, all of your folate will get tied up or trapped in methyl tetrahydrofolate, and this process will shut down, and once again you will be unable to convert uridine into thymidine monophosphate. And finally, if you're deficient in either vitamin, homocysteine levels will climb in the plasma because this sequence of reactions will shut down and homocysteine will not be metabolized into methionine. So now let's talk about why the last step in that complex sequence of reactions is so important. Let's talk about why it matters that homocysteine gets converted into methionine. And the reason you need to convert homocysteine into methionine via those reactions on the last slide is because methionine is necessary for synthesis of a very important structure called S-adenosylmethionine, or SAM. This is a cofactor that donates single carbon groups or methyl groups to other molecules, and S-adenosylmethionine is synthesized from ATP and the amino acid methionine. This is the structure of the amino acid methionine, this is ATP, and if you add them together, you get S-adenosylmethionine. And you can see that it has this one carbon right here hanging off of a sulfur group, and this carbon is ready to be donated to other structures, and that is exactly what SAM will do in many reactions inside of cells. When S-adenosylmethionine donates its methyl group to some other structure and then adenosine is removed, what you are left with is the amino acid homocysteine, and this is the structure of homocysteine on the screen here. Via that B12 folate pathway I showed you a few slides ago, homocysteine can be converted back into methionine, and this is the structure of methionine. And you may notice that homocysteine and methionine have almost the same chemical structure. The only difference is this extra methyl group, this extra carbon group on methionine. And that carbon group comes from that B12 folate pathway I showed you 2 slides ago. Once you have regenerated methionine, that can be converted back into S-adenosylmethionine, and thus this pathway continues so that cells have a pool of SAM always available to donate methyl groups to other structures. So just to summarize, from methionine you can generate SAM, and when SAM donates a methyl group, that yields a molecule of homocysteine. So you need to regenerate methionine in order to have a ready pool of SAM available to continue donating methyl groups to other structures. And the way that methionine is regenerated from homocysteine involves the vitamins B12 and folate. There is an enzyme called methionine synthase which requires vitamin B12, and it will add a methyl group to this sulfur on homocysteine and therefore create a molecule of methionine. The source of that methyl group is the molecule N5-methyl tetrahydrofolate. This methyl on N5-methyl tetrahydrofolate is removed to create a molecule of tetrahydrofolate. The big picture here is that you need both vitamins B12 and the vitamin folate in order for this process to occur, and if either vitamin is deficient, you will have levels of homocysteine in the plasma rise because you're unable to convert homocysteine back into methionine. So back to our big picture slide, which should make sense to you now. What we want to do is convert uridine into thymidine via the enzyme thymidylate synthase. This involves addition of one carbon to a molecule of uridine monophosphate. The source of that one carbon is the structure N5N10 tetrahydrofolate. When it donates that carbon group, it becomes dihydrofolate, and therefore dihydrofolate must be regenerated back into N5N10 tetrahydrofolate. The way that B12 comes into the picture is that N5N10 can be converted into N5-methyl tetrahydrofolate, and if you don't have B12, all of your folate vitamin will become trapped in this form, and it won't be available as N5N10 in order to synthesize thymidine. When B12 regenerates tetrahydrofolate from N5-methyl tetrahydrofolate, it converts homocysteine into methionine. Therefore, a deficiency of either vitamin results in high levels of homocysteine. So if you are deficient in either vitamin B12 or folate, you will be unable to synthesize thymidine. And because thymidine is necessary for DNA synthesis, this means you will have ineffective synthesis of DNA. And when you have ineffective synthesis of DNA, the clinical syndrome that develops is called a megaloblastic anemia. This is an anemia, as the name implies, with a low hematocrit. It's megaloblastic, meaning that the cells are large and therefore the MCV is increased. And then the classic finding on a peripheral blood smear is a hypersegmented neutrophil. I've shown you a picture of one of these on the screen. And megaloblastic anemias are caused by processes that lead to defective DNA production. The two classic causes are a folate deficiency or a B12 deficiency via all of the mechanisms I showed you on the last few slides. There is a rare inborn error of metabolism called erotic aciduria, which I discussed in some of the other biochemistry modules, and it can lead to a megaloblastic anemia. Drugs that interfere with the synthesis of thymidine and DNA can cause a megaloblastic anemia. These include methotrexate and 5-FU and hydroxyurea. And then a megaloblastic anemia is also often seen among patients on the HIV drug zidovudine. This is an HIV nucleoside reverse transcriptase inhibitor. This drug mimics the structure of thymidine, and therefore it can interfere with DNA synthesis inside of cells. Now that we've talked about the role that folate and vitamin B12 share in the synthesis of thymidine, let's talk about the two vitamins individually, and we'll begin with folate. This is the structure of folate on the screen. You can see that it's a large molecule. You'll also notice that it has lots of oxygen and nitrogen molecules, and vitamins with oxygen and nitrogen in them tend to be water soluble, and that's true of folate as well. Fat soluble vitamins, on the other hand, tend to have mostly just carbons and hydrogens. So the difference between folate and dihydrofolate has to do with this ring right here. You'll notice that this ring has 3 double bonds. If you add 2 hydrogens, you remove one of the double bonds so that the ring only has 2 double bonds, and then you get the structure of dihydrofolate. If you add another 2 hydrogens, you get the structure of tetrahydrofolate. In that case, this ring only has a single double bond. The difference between tetrahydrofolate and N5N10 tetrahydrofolate, the molecule that we were talking about in the synthesis of thymidine, is the addition of one carbon here between the 2 nitrogen molecules. That's why it's called N5N10. And that one carbon right there is the carbon that is donated to uridine in order to synthesize the nucleotide thymidine. Folate is absorbed in the jejunum. This is where most of the water-soluble vitamins are absorbed, and you should know that there are increased requirements for this vitamin in pregnancy and lactation. In both of these settings, there's an increased cell division. This means there's more metabolic demand and you need more DNA and more thymidine. And very importantly, the lack of folate in pregnant women can lead to neural tube defects. This is a baby with a severe version of spina bifida called a meningomyelocele. It's got some of the spinal cord material protruding through the back, and this is what can happen among women who don't take enough folate when they are pregnant. The classic type of patient to develop a folate deficiency is an alcoholic. Alcoholics often have poor nutrition and they don't take enough vitamins, including folate. Also, for reasons that aren't well understood, there's poor folate absorption among alcoholics. Therefore, when alcoholics present to emergency room, they're often given something called a banana bag intravenously. It contains a number of vitamins, including the vitamin folate. You can also develop a folate deficiency when you are taking certain drugs that interfere with folate metabolism. And the three drugs that do this are phenytoin, which is a seizure drug, trimethoprim, which is an antibiotic, and methotrexate, which is a chemotherapy agent. When you ingest folate, it goes to the GI tract, obviously, and then the folate must get absorbed into the plasma. And phenytoin interferes with the absorption of folate from the GI tract, and that's how you can develop a folate deficiency when you are on this drug for seizures. Trimethoprim and methotrexate both inhibit the enzyme dihydrofolate reductase, so they interfere with that cycle we showed a few slides ago in which dihydrofolate is converted to tetrahydrofolate. Trimethoprim is an antibiotic. It's designed to inhibit the bacterial version of dihydrofolate reductase, but it has some effect on the human version of dihydrofolate reductase. Methotrexate is a chemotherapy drug. It's also used for immunosuppression, and it's designed to inhibit dihydrofolate reductase in order to suppress the immune system or kill cancer cells, and therefore obviously when you take this drug you can get a relative folate deficiency because you can't metabolize folate properly. Now let's talk about vitamin B12, and vitamin B12 also goes by the name cobalamin, and the reason it's called cobalamin is because it contains the element cobalt in its chemical structure. Vitamin B12 is a large complex structure called a core-in ring. This is a picture of vitamin B12 on the screen. You'll notice that it has cobalt at the center of the molecule. You'll also notice that it has lots of oxygens and nitrogens and even a phosphate group, and therefore it is going to be water soluble. Vitamin B12 can only be synthesized by bacteria, but luckily many of the foods we eat contain vitamin B12 synthesized by bacteria in nature, and one of the most important sources of vitamin B12 is in meats. As we talked about at the beginning of this module, vitamin B12 is very important for the synthesis of thymidine, just like folate. But vitamin B12 has one other major role in cells that is unique from folate, and that is in the metabolism of odd-chain fatty acids. Folate has no role in the metabolism of odd-chain fatty acids, but you need vitamin B12 in order to metabolize these structures. Vitamin B12 is required for conversion of odd-chain fatty acids into succinyl-CoA, which can enter the TCA cycle. If you are deficient in vitamin B12, you will see high levels of methylmalonic acid in the plasma as a result of ineffective metabolism of odd-chain fatty acids. We'll talk more about this molecule in a minute. The high levels of methylmalonic acid are believed to contribute to the peripheral neuropathy that is seen in patients who are deficient in vitamin B12. Myelin synthesis is ineffective in patients who are deficient in vitamin B12, and therefore a peripheral neuropathy develops, and this is unique to vitamin B12 deficiency. You will not see this in folate deficiency, and this is one way you can tell the two vitamin deficiencies apart because, as I showed you before, they both contribute to megaloblastic anemia, but only in a B12 deficiency will you see a peripheral neuropathy. The neuropathy that occurs in B12 deficiency is called subacute combined degeneration, and it involves the dorsal spinal columns. I talk about this in more detail in the neurology modules on the spinal cord. It results from defective myelin formation as a result of B12 deficiency. The exact mechanism by which B12 causes this is unclear. Symptoms are typically bilateral, and they're usually in the legs much more than the arms. It begins as paresthesias, but it can progress to ataxia, and this may present in a board question as a patient with falls. On physical exam, there's loss of vibration and position sense, and it can progress and eventually cause severe weakness and paraplegia, and this is how many years ago people could die from a B12 deficiency. Let's talk more about that unique role of vitamin B12 in the metabolism of odd-chain fatty acids. Odd-chain fatty acids are metabolized into a structure called propionyl-CoA. I talk about this in more detail in the modules on fatty acid metabolism. Through a reaction that requires the vitamin biotin, propionyl-CoA is converted into methylmalonyl-CoA. And then there's an enzyme called methylmalonyl-CoA mutase, which requires vitamin B12, and it converts methylmalonyl-CoA into succinyl-CoA, which can enter the TCA cycle. And propionyl-CoA is an important intermediate for the metabolism of many different structures. Cholesterol can also be metabolized to propionyl-CoA, so can the amino acids isoleucine, valine, threonine, and methionine. All of these structures can become propionyl-CoA and then go through this reaction to enter the TCA cycle. And you need vitamin B12 for this step, the conversion of methylmalonyl-CoA into succinyl-CoA. If you are deficient in B12, this step will be ineffective, and therefore you'll be unable to convert methylmalonyl-CoA into succinyl-CoA. Methylmalonic acid can dissociate from methylmalonyl-CoA, and therefore in a B12 deficiency, you will see the level of methylmalonic acid rise in the plasma. And a high MMA level or methylmalonic acid level is a hallmark of the B12 deficiency, and very importantly, this is not seen in folate deficiency, and this is one way you can tell the two vitamin deficiencies apart. Most water-soluble vitamins are not stored very effectively in the body, so you can quickly become deficient if you don't consume enough in your diet. Vitamin B12 is an exception. It is a water-soluble vitamin, but the liver stores years worth of vitamin B12. Therefore, it is extremely uncommon to become deficient in this vitamin simply from a poor diet. The way you can become deficient in vitamin B12 is through an autoimmune process called pernicious anemia. In pernicious anemia, there is autoimmune destruction of parietal cells in the stomach, and these cells are very important because they secrete something called intrinsic factor. You need intrinsic factor in order to absorb vitamin B12 in the terminal ileum. If you lack intrinsic factor, you'll be unable to absorb vitamin B12 and you will become deficient. And the condition is called pernicious anemia. The word pernicious means deadly because years ago when people got this, their neurologic dysfunction would progress and they would usually die from the condition. So let's talk about pernicious anemia in a little more detail. This is an autoimmune disease that leads to chronic inflammation of the gastric body. This is a picture of the stomach here. This is the pylorus and the antrum, and this is the fundus, and this is the body of the stomach where the inflammation occurs in pernicious anemia. This condition is more common among women, and it has a complex immunology. Antibodies have been described in pernicious anemia against parietal cells and also against intrinsic factor. For these reasons, it's often held up as an example of a type 2 hypersensitivity reaction where antibodies attack tissues in the body. However, it's more complicated than just antibodies. There's also evidence of autoreactive CD4 T cells and other immunologic processes occurring in pernicious anemia. Like many autoimmune diseases, it's associated with certain HLA antigens. In the case of pernicious anemia, it's been associated with a number of different HLA-DR antigens. If you want to understand this more, watch the module on transplants in immunology. And then finally, because there's chronic inflammation, there's an increased risk of gastric adenocarcinoma among patients who have pernicious anemia. Besides pernicious anemia, there are other reasons you can become deficient in vitamin B12. If your ileum has been resected or it's dysfunctional, such as in Crohn's disease, you can be unable to absorb vitamin B12. If you lose intrinsic factor from the stomach, for example, if you've had surgery for obesity in the form of gastric bypass, you can potentially be unable to absorb vitamin B12. And then finally, a very rare cause that they like to ask you about on step 1 is the Helminth infection, Diphyllobothrium latum. It's a tapeworm, and you get this tapeworm from eating infected fish, and the tapeworm consumes vitamin B12, and it can make you deficient in vitamin B12. There are a couple of diagnostic tests that are used to detect a B12 deficiency. The first one, obviously, is to detect a low serum level of vitamin B12. If the serum level is borderline, you can measure the level of methylmalonic acid, and if it's high, that suggests a B12 deficiency. If you suspect pernicious anemia, antibodies to intrinsic factor are often measured. And then finally, there's a historical test that's not used much anymore called the Schilling test. This used to be the classic diagnostic test for pernicious anemia. In this test, patients are administered oral radio-labeled vitamin B12. In addition, they're given an intramuscular injection of B12 in order to saturate the liver receptors. As a result, if the patient is able to absorb the oral B12, the radio-labeled B12 will spill into the urine because the liver receptors are all saturated. So if you see radio-labeled vitamin B12 in the urine, that means the patient has the ability to absorb vitamin B12 by the Schilling test. If the radio-labeled B12 does not appear in the urine, you can then repeat the test by giving oral intrinsic factor and see if this corrects the problem. If it does, it means they have pernicious anemia and they're deficient in intrinsic factor. So as I said before, pernicious anemia gets its name because years ago, if you had this type of anemia, the neurological problems would progress until you would die. Luckily, nowadays it's treatable with a liquid injection. So even if you can't absorb vitamin B12, you can be given an injectable form of the vitamin to satisfy your body's requirements. It's usually given subcutaneously or intramuscularly, and you usually see an increase in reticulocytes as the patient begins to respond to the B12 injections. And that concludes our module on folate and vitamin B12.